Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome everyone to the Genetics Podcast. I'm excited to be here today with Ananth, Sridhar, and Sun-Gou Ji from BridgeBio, two leaders who are driving innovation at the intersection of rare disease drug development and human genetics. Ananth is the COO of BridgeBio's Cardiorenal programs through their subsidiary companies Eidos Therapeutics and Calcilytics, where he leads two of the company's most advanced therapies. And in his career, he's actually, I think, taken four therapies all the way through FDA approval, which is pretty exciting and something I think we can all aspire to. Sun-Gou is VP of Computational Genomics at BridgeBio, where he's leading a team that's focused on target validation and discovery through large-scale human genetics. Before BridgeBio, I understand that he worked on the Million Veterans program at 7 Bridges. We've actually had a previous guest from MVP on the podcast, so go back and check that out, and I'm sure we talk about some of Sun-Gou's work. But we're going to have a conversation today about rare disease drug development, BridgeBio, and both of these two's path into where they are today. So thank you both for taking the time to join me.
Thank you, Patrick, for having us.
Great. I want to start with BridgeBio's unique hub and spoke model. It's really important to this whole episode, and I think it's a really interesting model in general for how we do drug development. So maybe, Ananth, let's start with you. If you could just talk a little bit about what that model is and how it works for people that aren't familiar with BridgeBio.
Sure, Patrick, thanks for having us on. And the hub and spoke model is something that was an incepting thesis behind BridgeBio, and it has a operating feature and a financing feature that helps enable its operational success, or at least its validation in our current state. And the operational model feature is that we have decentralized our functional teams, being that we have teams focused on an individual molecule. And that enables technical expertise at the depth of the molecule and the indication that we hope to address with it. It enables relationships with the key thought leaders in the field, enables relationships with the advocacy organizations. And that integration and that level of program depth is something that's unparalleled, at least in my experience in the industry, because if you're stretched across multiple molecules or even therapeutic areas, you don't tend to form those deep, lasting relationships or the level of insight that it takes to advance thought and innovation. And then from a financing perspective, the other element of the hub and spoke model that I think is really critical is that it helps de-risk our investments. A success and failure in program A does not portend any odds of success or failure to program B and program C and thereon. And the structure therewith is that it enables an optimization of a risk return profile that enables a lower cost of capital and a higher propensity for investment to advance our research. And so we have evolved the model with time. I think where we stand today, we have dedicated, small, focused, lean program teams, and then we centralize some of the resources that don't require molecule level knowledge. And those are, some functions that you would expect, some things like human resources and finance and legal. And then at the level of the program, we have the depth and expertise from our functional experts.
Yeah, super. That's a really great explanation. And I've watched previously a great YouTube video from Andrew Lowe from MIT, who's one of the I think pioneers behind this model, and I'll put the link in the show notes to encourage people to watch it, because for the mathematicians like me, it gives a nice mathematical explanation of why this is a nice way to structure it. But maybe Sun-Gou, this is good.
Just wanted to add one more thing to it. It's
Yeah.
I think this model also, that's maybe underappreciated is that it allows for like funding towards some rare diseases that would not be possible to be funded through unless we have this hub and spoke model, right? A lot of these rare diseases happen to be genetic, and we know that genetic disorders have higher chances of success. But despite that 2x increase of success, just a single program just like succeeding is still very low, right?
Yeah.
So if you try to fund a single program on a rare disorder, it's very difficult to get funding. But if you bundle them together, right, then it becomes this mathematical problem. So how many programs do you need to do to actually have a certain probability of the whole portfolio making it rather than a single program making it? So this new way of funding that BridgeBio and Andrew Lowe has pioneered allows this kind of funding for smaller indication that just may have smaller upside, but we take the risk into account rather than just the upside, which is like peak sales.
Yeah, interesting. So is it right that you may know that one program in and of itself may not be likely to be a commercial blockbuster, but if you look at the genetics and you look at the unmet need in the patient population and the risk is significantly lower, then actually you can build a blended portfolio that has a small number that may drive the peak sales, but you have some, yeah, I guess to use a baseball analogy, more base hits that you can help patients, you can generate money, and they're going to be net profitable to the enterprise overall, but they don't have to in and of themselves be a blockbuster. Is that the right way to think about it?
Exactly. So we would want a mixture of things, right? You want those blockbusters potential with higher risk, like smaller things that may lower risk, and the risk could come from different ways. There's not a program that has no risk, right? We just want to make sure those risks are well distributed and kind of this uncorrelated risk, and these shots being independent is important for us as a portfolio level.
If we think about your work in genetics and target discovery, where do you think about what belongs in the hub and then what belongs in each of the spokes? And how do you think about depth in one of the particular areas like ADH1, for example, that Ananth works on versus building a core hub that can help you figure out what that next target is and then work closely with that functional team to chase it down in a lot more detail? Because I noticed Ananth said the molecule level rather than the target level, which maybe is an important distinction.
Yeah. So the distinction between hub and the spoke always kind of gets blurry all the time, depending on the question, depending on the target and things. But usually when we're talking about which target to go after, or are we interested in kind of building a clinical program of this on this target, is the target selection and actually starting a program is all central. So, and we have a very respected investment committee that kind of gets together to kind of make a decision on which programs do we like this risk in the context of the other risks that we have in our portfolio, and make that selection. And when that kind of diligence process is happening, that's when the computational team comes in to do a lot of the vetting against the target or against the molecule, against the therapy, using all the genetic information as well as the human biobanks on all the multi-omics data sets that's available right now to kind of make that case about a program being a BridgeBio program or not. But once the program is a BridgeBio program, I think it depends on whether especially if it's near clinical, that's usually when we spin them out as independent entities. But until they're clinical, we usually kind of incubate them a bit in the central space to make sure that we have a good chance of getting to a DC, and then we start building a separate team once that happens. But yeah, it's very different by indication, by the opportunity that we're diligencing. If it's a huge opportunity, I mean, like if it's a very big, clinical trial that's needed, we might try to spin it off a bit early. If it's not, and so there's a lot of things that goes into when do we spin things out as a spoke versus incubate further under BridgeBio.
Yeah, that makes sense. Ananth, maybe you could talk a little bit about ADH1, that program. I think that's been an interesting journey as well of repositioning from one indication into another, and what you learned there maybe about following that molecule, where the biology leads you, and that program. I think it may be interesting to just pull the thread on that and help people understand a little bit more about the work that you do at a, at a micro level in one of these programs, then we can zoom back out to the big picture.
Yeah, it sounds great, Patrick. And I'll riff off of some of Sun-Gu's words here, which takes us back to the beginning of how did we identify this program, and really it does start with like the target, like Sun-Gu mentioned, and then it evolved into our selection of the right molecule. The target here is the calcium sensing receptor, which is a G protein coupled receptor. It's a class C GPCR that's primarily expressed on calcitropic tissues. So we see the expression profile most highly present in the parathyroid gland and the kidney. And in ADH1, the calcium sensing receptor is overactive or oversensitized to extracellular calcium. And as a result, it leads to overexcretion in the urine and low blood calcium chronically for patients. How we came across this target was actually through a couple of means. One is a paper that was published in the Journal of Bone Mineral Research in 2019 that exposed a small cohort of 5 patients to a intravenous calcilytic molecule to try to see if there's a proof of principle that this thing could work in antagonizing the receptor selectively, may be able to restore calcium homeostasis. There was signals of early success in that cohort, but it was not necessarily the best designed study, nor was it the best necessarily molecule for the indication. And then the second element was actually close collaboration with leading academic scientists at the National Institutes of Health, who had not only published this work, but also been amenable to our discussions with them to learn more about the receptor biology and patient care. So we translated a lot of those early insights into our development program of encaleret, which is now in its phase 3 study in autosomal dominant hypocalcemia type 1. The hope there is that we can provide these patients with an option that not only increases their blood calcium into the normal range, but also reduces urine calcium. So theoretically redefining the calcium homeostasis set point for these patients, which cannot be achieved currently with conventional therapy. So conventional therapy for these patients today tends to be calcium supplementation and or active vitamin D, which can increase blood calcium certainly and resolve some of the symptoms that are present with hypocalcemia, but tend to exacerbate the hypercalciuria and the renal effect. And so we hope that encaleret may provide an option that could address both the renal and the blood aspects of calcium homeostasis and calcium regulation. And the calcium sensing receptor is turning out to be an important regulator of this important electrolyte.
What's been the biggest challenge of that program along the way, would you say? Because every rare disease program is probably a little bit different, but they all probably end up rhyming and having some common themes, right? What was, before we get into some of those themes, what's the biggest challenge there?
Yeah, I'll start here. And we partner closely with Sun-Gou and his team on a couple of these important elements. I think one of the challenges is that we are first in this field in a few ways. We are the first to investigate this class of molecules in this patient population, and we're also the first ever to conduct a sponsored clinical study in this patient population. And so that requires a couple of elements that are just challenging for human behavior. One is to think deeper. If you have signs and symptoms of ADH1, historically, there's been limited actionability of genetic testing to identify the gain of function variants. And then also, and I'll pass to Sun-Gou on the technical features here, but identifying gain of function variants is not trivial.
Right.
And it requires depth of expertise and clinical features often to really identify gain of function variants versus loss of function. And so I think one of the elements that I would classify as our learnings as probably some of the challenges we are facing is really identifying the patients and enhancing diagnostic suspicion in the field, such that if there are candidate patients or patients who are suffering or families who are suffering from ADH1, that a proper diagnosis is made. But Sun-Gou, I would love to, for you to elaborate not only on the prevalence estimates that our team has worked on, but also
on our abilities to identify gain-of-function variants of the
Yeah,
calculus.
yeah, for sure. So we recently published a paper on the American Journal of Human Genetics regarding the kind of allelic series that we observe on the calcium sensing receptor. And it's an interesting gene because it has both loss of function associated disorder, which is familiar hypocalciuria, hypercalciuria, and the gain of function, which is the disorder that we are treating, is autosomal dominant hypocalcemia. And as you can expect, loss of function leads to hypercalcemia, gain of function leads to hypocalcemia. And actually, if you look at the GWASs of calcium, like CASR, is one of the biggest hit that's been observed consistently across GWASs of calcium in humans. So it has a very nice allelic series. So what we think as geneticists is that if you can control this, it's not just an on and off switch. It's actually a good kind of a gradual lever that could be used across not just these very severe forms of autosomal dominant hypocalcemia, but any other kind of common complex form of calcium-related disorders could be used. But our first question was, of course, because we are going for the monogenic disorder first, is how many people actually carry a gain of function variant. So the simplest thing you could do is go to the UK Biobank, count the people that have these known gain of function variants first. And there are quite a few, and we were surprised to see so many of them. So we try to validate it across all of us and the MGB biobank, as well as all the other biobanks or genetic information cohorts that we had access to, just counting up the number of pathogenic variants that's been observed. And the number was always quite consistently at like 4 in 100,000. But then the second question was like, okay, since we know so much about, there's actually lower penetrance, right? That's what all these genetic analysis have been showing, is that there's definitely higher genetic prevalence of pathogenic mutation carrier, but penetrance may actually be lower than what we've learned in high school and actually recent biology classes. So we actually went into all of us and UKB, where you could actually look at patient phenotypes. And what we found was that despite less than half of them having some sort of a diagnosis code related to ADH1, most of them had low serum calcium and other phenotypes that's associated within the kind of phenotypic spectrum of ADH1. And we also identify a bunch of other symptoms that may not be classified as classical kind of ADH1 phenotypes, but are still within the kind of phenotypic spectrum of CASR function. So that led to this publication, but it's still hard to find these patients from the other side. So what we've been able to do is just to kind of genetics first patient finding, and we've been able to get to a pretty confident number of how many people have mutation that actually have symptoms that needs treatment. But that's when you know the genotype. But the real world problem is, how do you enrich from like the phenotype, which is the real world data, to people that have the genotype from all these very benign, seemingly benign combination of clinical symptoms. So that's still ongoing work that we're trying to figure out. We know how to do it from genotype first, and then how do we do it from phenotype first is still an ongoing project that we're working on.
Yeah, that's really interesting. How often, when you look across these, moving out of ADH1 for a second, how often do you find that you have an underdiagnosis issue in the diseases you work in, and then how often do you find this penetrance question? Because it almost seems like a law of gravity a little bit, where there's always more patients than you think, but there's also always less penetrance than you think the more and more that you find. Is that pretty much a assumption that you should start with across the board?
I think so. I don't think I can name a disorder that it's. I guess one disorder that's pretty clear where the observed penetrance and the observed prevalence is similar is achondroplasia, where it's actually driven by a single variant and the phenotype is very clear and diagnosis very clear. But most of the rare disorders, I think there's many publications that precede us and also coming out very recently, the ones that precedes us is like Hugh Marcus's work on like monogenic stroke that they've published on NOTS3 or like 4 times the expected number, and also Caroline Wright's work on like neurodevelopmental disorders, a huge increase in people that carry the pathogenic variants, but then actually it's smaller. So and you could name many others that came out recently. I think there's one on NF1 actually from UPenn, Alport also, like from a recent paper from Geisinger. But so this is like everything that we know of almost has this. But then that's why it becomes even more important to quantify that difference, right? And that's why we're like, I think every rare genetic kind of companies or advocacy always say that there is an underdiagnosis based on the genetic prevalence, which is always true. But the quantifying the difference between the genetics and the actual phenotypic kind of spectrum, and of that phenotypic spectrum, what requires treatment is also a different problem. Right. So we feel like there's a ceiling, but then there's some blur between which phenotypes associated with carrying a pathogenic mutation in this gene actually needs treatment. That would need a lot of discussion with clinicians at treating these patients. Because even though you have the exact same gene kind of burden, when do you treat or not may be very different per person, and how a clinician makes a decision based on all this complicated information will need a lot of discussion.
Yeah, that completely makes sense. I want to maybe pull on the thread of this, of you being first in class in this particular disease. And there's a general challenge or issue, I'd say, that we have around herding in rare disease drug development more broadly. You tend to see dozens of programs going after the same target, and then there's, thousands and thousands of diseases where there's nothing going on. On the one hand, they may be the best targets, right? So it's explainable in some sense, but it also creates, a challenge where you have too much resource going into one area and not enough going into another area. How do you all think about this internally in terms of what you prioritize and how you focus on going after something where you really understand the target and think you can do something there, but other people may be going after it too, versus taking a risk on some new biology? Maybe Ananth, if you're happy, we can start with you on this one and then go across to Sun-Gou.
No, it sounds great. Yeah, and Patrick, this might be something familiar to you and your team over at Sano too, which is that the pioneering endeavor to go first in class and endeavor to investigate a program that's the first of its kind comes with the downsides of finding the patients, recruiting the studies, and establishing regulatory precedents, right? And those things tend to take time, effort, and considerable risk. But what we can do to de-risk the elements that I described is mapping the science, which is genetics first to the biochemical feature to the phenotype. And if we can take it one step at a time in our research efforts, which is especially in the clinical setting, if we can design the right experiments early, improve the concept, even on that biomarker that precedes the phenotype, we can start to de-risk some of those operating features that come up as we accelerate these programs. And then in terms of an operating philosophy, going first or second, I'm personally of the mindset that there's not really that much of a human value or a human impact that being the sixth PD-1 or the sixth GLP-1 actually is going to deliver. I really think that there are certainly economic features, there may be convenience features that can be enhanced certainly for a patient experience, but I really think if we want to leave an impact, a lasting impact on biology and on human health, to make those revolutionary changes versus evolutionary changes, it does take some of these bets. And the way that we can de-risk is heavily is follow the science, but especially with monogenic disease, where in many cases nature's run the experiment for us. And in this disease in particular, just go back down the rabbit hole. ADH1 is the flip of this disease is a condition called familial hypercalcemic hypocalciuria. These are caused by loss of function variants to the same receptor. So we almost have a nature experiment of the extreme model of calcium sensing receptor negative modulation. And so we can study a lot from parathyroid hyperplasia that can occur to the downstream renal biology from patients who exhibit loss of function variants to this receptor. And so I think the technically de-risking these challenging programs where we are pioneering some of the science can help obviate or at least lower the threshold or the ceiling on risk on the operating features.
Sun-Gou, any thoughts on that from your perspective in terms of how you focus on the work that you do?
Yes, so I guess Ananth touched upon the science, of course, and the patient unmet need. I could also talk a bit about how we think about it on the economic side. So we also released a white paper recently called The Feasibility of Rare Disease Drug Development. And that's basically, if you read through it, you can kind of get a sense of how BridgeBio thinks about which programs to pursue. And it all centers around the risk-adjusted NPV, right? And so one of the reasons that hurting happens in rare disease drug development is that people focus on peak sales. You want to make sure that the peak sale hits certain numbers. And if you do that, and if you look across 10,000, genetically driven, monogenic disorders, there's very, very few that actually cross that peak sales number that we know. And that's one of the reasons. And if we don't, I think one thing that actually that the white paper that we put out try to argue for is that we should, not just think about peak sales, but think through RNPV. And because peak sale only considers the potential outcome of this drug and ignores actually the potential cost to get there, right? And in contrast to common diseases like IBD, if you make a drug there, RNPV is not relevant because the cost of developing a drug will be negligible in the context of the large fruit at the end. But in case of the rare diseases that we work on, actually comparing the size of the fruit that will bear with some probability against the expected cost is actually critical, and some are just not economically viable, whereas some are. And that's why the opportunity Ananth is kind of in Coloret, or the calcilytics in Coloret, where Ananth and his team has pulled off one of probably the cheapest drug development program ever, will just not have happened in other places. And this is one way I feel like we can, as a group making drugs, can try to not just think about peak sales, but think of what goes in and what goes out, and in the context of time to make decision a bit more data-based.
Yeah, and just to define for everybody, net present value is essentially taking all of the future cash flows and giving it some discount going into the future, right? Because you don't have it all today. And then if you were to pull it back to the present, almost like the reverse of a loan, or a loan, I guess, is the conceptual idea. If you had all that money today, what would it be worth? And compare that to what it's going to cost, discounting the time that takes. It's a nice way to think about it. And I understand why people focus on peak sales, because it's a little bit simpler, but sometimes simple isn't always the right answer, right? You have to go one or two steps beyond. Maybe, Ananth, you can give your perspective on that. I'm interested in the hallmarks of what make a less expensive or faster program. Is it down to the type of molecule that you use? It strikes me as like an RNA, for example, might be a better choice than gene therapy in many cases. But I'm curious to get the couple of factors that make for a program that can be low cost and fast and unlock some of those rarer populations.
Yeah, that's a fun question to answer, because it helps tether a lot of the conversation we've had throughout this discussion. So good described it very astutely, which is peak sales is just one element of an NPV, or a net present value model. But there are two other levers that I think are critical, which are time and on cost. And if you can drive down time to achieve approval and sales, and you can drive down cost to get there, you can optimize or maximize an MPV, keeping sales flat. And I think that's a really important element of how we can do that. I think in rare disease, you have to, especially in some of the smaller markets where we play. But the other element of it is that we can be very clever and thoughtful in our clinical research experiments, wherein we don't have to enroll 1,000 patients at 900 sites globally, where the cost can really start to escalate. I think in genetic disease, we can establish a predictive biomarker that tends to be downstream of the protein that is aberrant. And if we can demonstrate a profound impact on that protein, we can drive down the cost and the time to development such that we can be more nimble and optimize our net present value. And why I say this is a fun question to answer, and it goes back to some of the comments we made earlier, which regarding that the ability to go after these very rare diseases, these N of 1 type diseases with our hub and spoke model. You know, if we can make MPV positive bets, meaning that the modeled cash flow with risk baked in and discount factors baked in are still on the positive side of an investment, it's worth doing. And so it makes us and allows us to have those decisions made on a more simple basis than to say, how do I fill a $2 billion revenue gap that I anticipate in 2 years, right? So I think, you know, that's why it's a really a strong feature and perk of the BridgeBio model, that we can go after these very rare diseases and optimize time and cost and the right teams so that we can advance science very nimbly.
Yeah,
You know,
really.
we released a small model with the parameters that you could tweak around on the white paper that we've published. So if people are interested, they can go in and try to maybe move the parameters that matches whatever drug that they're working on and see. And if they're interested, have any questions, yeah, they reach out anytime.
Yeah, I love that. I'll put a link to it in the show notes because I think that's super cool. I will personally be playing around with that because at least I'm interested. I'm sure many of the listeners will be as well. I actually want, yeah.
Got to answer one part of your question that I wanted to just touch on, which is you asked about modality, which is, you know, gene therapy versus RNAs versus mAbs. I really think that being agnostic to those technologies is what will enhance and unlock the best medicines for patients. If you, I think, commit to using a baseball bat when a putter can be the right option on the green, I think, you know, it doesn't make sense. But I really believe that we're actually kind of in a cool phase for small molecules because we have some pretty advanced computational tools that can really model receptors. We have really nice cryo-EM experiments that we can run to look at how a receptor presents itself. And we know there's several GPCRs that are present in our system that have extracellular binding domains or transmembrane binding domains. So small molecules do have an option. I think a cool renaissance coming, and I also believe that, you know, mAbs, where I've spent a lot of my research efforts, continue to be kind of, you know, the nature's medicine, and I think there's a lot of untapped opportunity there beyond the next advent of technologies like mRNA and gene therapies.
Yeah, I completely agree with you on that. I think people think precision medicine and they automatically go to RNA and gene therapy, but really it's about understanding the underlying biology and targeting it in a more precise way, right? So I think it's an absolutely great point to make. I actually want to go just very briefly down memory lane for you both to give people a sense of some of your past experience and how you got here. And Sun-Gou, I'd like to start with you because we have a lot of people who, like you, work in target discovery, computational biology, statistical genetics across academia and/or pharma and biotech. When you're at Seven Bridges, you built software that was used by lots of groups like yourselves to do statistical genetics and genomics across drug development. But now you're on the other side of the table, and you probably are making decisions about what do I build internally, what do I partner on, how do we do this based on everything that we've learned and all the expertise we had. And I'm just curious to get your perspective on sitting in a place like BridgeBio. How do you think about what you do to be competitive and to do something that you can't just do by accessing UK Biobank. You know, there's a threshold that we have to cross to find something that's novel that isn't in the usual suspects. So how do you think about differentiating there? Is it data? Is it internal software? Is it choosing the right partners? How do you think about doing that?
Well, that's a very difficult question, Patrick, and I'm sure everyone's struggling with it right now. So let's start with data. Of course, the data is key for whatever we do, but also, you know, we are not at the space where we're kind of actually able to, let's say, fund the UK Biobank exome sequencing or that type of a big pharma level spending. So we have to be very focused on what data to pursue. And of course, the UK Biobank, the All of Us, and a lot of these more openly accessible data sets have been hugely helpful. But for us, we really try to focus on like actually getting access to the ones that are obvious, but also the ones that we care about, which is rare genetic disorders. So that's why the Genomics England is a good partner for us, and as well as genetic testing providers like Invitae and GeneDx. Those are the places where the data is enriched for these type of kind of genetic disorders that you would not get that kind of a sample size even if you sequence 2 million people of the common population. So those are the places that we are focusing on, kind of data access. But then the second point about how do we enable, I think that's the part that everyone's really struggling, because back then in 7Bridges, I think it started off as kind of this graph kind of enabled kind of cloud genomics platform. And when you and me were doing our PhDs at the Sanger, running things on the HPC was the standard, right? And people had the idea that moving to the cloud will happen eventually, but there was also a very, people were scared of it, right?
Yeah,
what happens
I remember
to
them being scared of it. Yeah.
Yes, and that's where sort of the niche of 7Bridges happened. And also it was almost impossible to run anything on AWS directly. So there was a need for a platform to help you run things on AWS directly. But then now, I think most things can be run AWS directly, right? It's so much easier to run things there. But then the problem now that becomes is like every one of these data kind of owners have their own trusted research environment that you want. So now it sort of becomes not the choice of us kind of designing a platform that we want to do our work on. It's actually learning different platforms, figuring out how to pick things out, and figuring out how at the different place where you could kind of merge them together. So I think my job has become not less of designing. I think before it was when we were able to download the whole of UK Biobank and run place and do things ourselves. We were thinking about optimizing where is the platform that we could actually do most of the things, download the things and do that. But now I'm mostly just figuring out how to piece these different TREs together to make things work as soon as possible. And I don't think honestly have seen a solution here. And I think this is something that everyone is struggling with. And I think many people that are doing this across many companies are discussing a lot on how to solve this.
Yeah, I think you hit the nail on the head on that. Ananth, you, as I mentioned in the intro, have been a part of 4 FDA approvals. You spent a lot of time at Regeneron before you came to BridgeBio, which is one of the examples that Sun-Gou mentioned that is also very deep in genetics, but in a slightly different way. I'd love to get your take and learnings on what you've learned about drug development from your time there and your time now at BridgeBio for the audience.
Sure. Yeah. I mean, in short, it's humbling. It's the easiest one-word descriptor. The work from Regeneron that enabled some of the fortune of being part of teams that advanced programs through FDA approval helped eliminate a couple of things. One is that new medicines are transformative. I think a lot of the patient circles that we get to be exposed to, one of the most challenging questions that are faced in any disease, particularly rare genetic, but I think in any marketplace is why me? Those are the hardest questions for industry players, scientists, clinicians to address. But I think all of the field, the colleagues I've been privileged to learn from and work with want to turn that why me into what's next.
Yeah.
And that's a kind of a guiding principle that I've really learned a lot from and adopted in my mindset here. And it goes to show even within Colorado, we didn't want to leave our program with just the ADH1 opportunity and the research we're doing there. We have also advanced it in a broader condition of chronic hypoparathyroidism, which was enabled by a lot of the partnership we've had with Sun-Gou and his team on the genetic side and the allelic series that we built the evidence around, which has enabled and informed a clinical experiment in chronic hypoparathyroidism, where we show that encaleret could be an orally administered agent that can really transform calcium homeostasis for a broader condition too. So for me, it's really listening to these patient anecdotes and trying to glean the impact that we can have or the unmet need that does persist. And that's been enabled through learning from great mentors at Regeneron, Genentech, and BridgeBio Lake.
We have a couple minutes here, and when I have two very smart people, I like to ask my favorite question, which is some area of precision medicine or genomics that you're really excited about right now, because it helps me figure out who I go to talk to and what topics I learn more about. So Sun-Gou, I'll start with you. What's the thing right now that you're particularly interested in?
Yeah, so I think you gave me this question and I kind of answered it like a couple of months ago, and it was newborn sequencing. And I think you've already spoke to the biggest players there on your podcast, but I do want to kind of reiterate why I feel this is like very important, both for patients and healthcare, as well as for the data that will be generated by doing this type of unbiased kind of screening or data generation across the population. So, I have a 9-month-old girl who has recently been diagnosed with a rare genetic disorder, and we were, I guess, lucky enough to be living in Boston, where our pediatrician knew to refer us to a specialist, who then quickly sent us to Boston Children's, and we got the best care possible. And within a couple of days, got a diagnosis and started treatment right away. And the nurses and doctors at Boston Children's were so helpful. They were super supportive, full of empathy, and we are so grateful for our care team. And I think I've really experienced like the world-leading medical care within the U.S. medical system, as it should be. I think we do talk about the bad things most of the times, but when it works, it was almost like beautiful. But also, I do understand that I was pretty lucky to being diagnosed so quickly. And one thing after this experience, when I was looking back, and one thing I regret was that I actually convinced myself that I didn't want to get my daughter sequenced when she was born. I used the exact same logic against newborn sequencing to convince myself that I'd be overwhelmed with this information. But looking back, I do feel there was some element of laziness on my end. And like, just if I analyze their genome myself and have the information of the handful of genes that might potentially, carry some bad variants, and I know penetrance, so I wouldn't be overreacting like many others would. And could I have reduced the search space of what I should prioritize looking for, right? And just thinking about, would it have been possible that maybe I could have picked up her symptoms a bit earlier? And of course, earlier treatment is always better. But with the benefit of hindsight, I think I would have been possible to catch this earlier. So despite like the medical system being like super, we've been lucky enough to be treated. And I think this is the best case scenario for the medical system right now. I think we have the technical ability, and that's the case, right? This is all possible now. The technology, these assays are so accurate. Interpretation needs some, improvement, but the only way to get that improvement is just by doing more. Yeah. And like, Wendy Chung from and like the Guardian and all these things, but also of course the UK, I mean, leading the way is really good to see. Like just like by identifying the cases like my daughter a bit earlier, and just kind of reducing or prioritizing the search space for the parents for things that they should be looking for, but not get alarmed. I think it's all like education and operations things that may be harder than technical things, but I feel like, at least for me, that would have been super helpful if that information was there. And in addition, the data generated will have huge value for genetics research to kind of develop drugs and discover new ones based on this true, like, unbiased information from the population.
I'm so sorry to hear of all that you've been through in the last 9 months, and thank you for sharing that. It's obviously every parent's worst nightmare to have to make their way through that kind of process. But I think what you say is spot on. If we can enable everyone to have access to the kind of expertise that you have or others have, and shorten that time to diagnosis and help parents know what to focus on, I think that there's a huge amount of opportunity to get way ahead of some of these diseases. And maybe, Ananth, this is a good segue. I know that's a very difficult one to follow, given that Sun-Gou is speaking from the heart on this, but I'd love to have your perspective as well on something that you're focused on and really excited about right now.
Yeah, it's a profoundly hard one to follow. It hits hard because human health is just so important to everything we do. I think my kind of what gets my antenna up and just me excited about maybe the future of genetic medicine specifically is something I heard in one of your prior conversations, I think with Vanita Agarwal, which was how can we transform preventive medicine, which is really important, into predictive medicine, and that becomes more human. Rather than saying you have a risk of adopting or developing these conditions, can we actually go one step deeper and say, here's your odds of that, and even in the option of medicines, here's your odds of responding to each of these therapy options that may be available to you. As we know, no one drug is great for everyone, and so if we can turn that around and say which drug is right for me, or which medicine is the best for me, I think that would be a frontier I think I'd really be excited to be a part of.
Yeah, I love that. Thank you both. And I think those are connected together in a very clear way. So I appreciate all the time you've given to us today and all the great expertise you bring. So thank you both for joining us.
Thank you, Patrick.
Thanks, Patrick.
And as always, thank you everyone for listening, and we'll see you next time. Thanks as always for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sanogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sanogenetics, on LinkedIn, or reach out on Instagram @The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.